It's quite a turnout. That's good. And actually, Aaron's violated our dress code here.
Sorry.
He's a little too fancily dressed, but we'll accept that. Anyways, I don't want to take his time. I think you all know Aaron, who's a National Library of Medicine Predoctoral Fellow in the BCB track. He received his Bachelor's of Science in Biochemistry from Pacific Lutheran University in Tacoma, and after graduation did some bench work, decided that he liked computational work better, and that led him to join our program. And he has established a mentorship relationship with Dr. Michael Chang, who is both an informatician and ophthalmologist and is focused on retinopathy of prematurity. And so today Aaron is going to defend his dissertation proposal entitled Machine Learning for Disease Detection and Prediction in Retinopathy of Prematurity. So he'll present, and then, well, since it's his proposal, the audience will ask questions, and then there may be additional questions afterwards from his committee.
Awesome. Thanks, Bill. Thank you, everyone, for coming to attend. So, as the title suggests, it's going to be about machine learning, but more specifically, we're going to be talking about some different sort of database methods that we can use, as well as deep learning techniques that we can use to inform physicians as to whether or not a child, in this case, is likely to progress or regress in terms of their disease severity. So just some quick acknowledgements. As Bill mentioned, my advisor is Michael Chang, but I'd also like to acknowledge the other members of my DAC, Dr. Songmez, Dr. Kalpathy-Kramer, and Dr. Campbell, as well as the faculty, staff, and students within our department, DMICE, and the National Library of Medicine for the training grant that they provided to me. So retinopathy of prematurity is this potentially blinding disorder that affects premature children that are born prior to 31 weeks of gestation and that weigh less than 1,500 grams at birth. And it's actually one of the world's leading causes of childhood blindness. Now, of the children that actually develop ROP, only about 15% will move on to this treatment-requiring phase of ROP, but they need to be treated within the next 48 to 72 hours because the disease is so aggressive. Now, unfortunately, we have a severe shortage of ROP experts just globally. So like a lot of the time, these children who come in for screenings, they'll get screened maybe once every week or two, but in that time, they can often progress to the treatment-requiring stage of ROP and actually begin to lose vision within that between screening periods there. So before I move any further into describing ROP, I think it's important that we kind of orient ourselves to what it is we're talking about in general by looking at the eye here. So this is a retinal fundus photo of an infant eye, of a normal infant eye. And there are three major features here. So the first is this circular object here that you see, and this is called the optic disc. And the optic disc is basically collecting all of the raw image data that's collected by the photoreceptor cells of your eye, and that image data is then sent to the visual cortex of the brain for image processing and helps make everything make sense. Then radiating outward from the optic disc, you'll find the retinal blood vessels. And these are just supplying oxygen and nutrients to the photoreceptor cells as well as other supporting cells of the retina. And so I keep mentioning these photoreceptor cells, and in your peripheral retina, you'll have these cells which are called your rod photoreceptors, and those are responsible for seeing in lower light conditions and responsible for your peripheral vision. And then you also have your cone photoreceptor cells, which help you see in better lit conditions and are also responsible for seeing color, and these are concentrated in this area that we call the macula. And because they're like so concentrated in this area, this is actually what gives humans great visual acuity. So you can imagine that having damage to either the optic disc or the macula would be cause for concern and could actually severely degrade someone's visual acuity. So here's an eye with retinopathy of prematurity. And the first clinical finding of ROP is that you'll notice that there's this area of avascular retina, meaning that the retinal tissue has not fully grown out into the eye. And the way that we actually stage ROP is by looking at this junction that occurs between the vascular and the avascular retina. And to do this, we have 5 different stages. So the first stage is, we just call this, the junction there looks like a simple demarcation line. But in stage 2 ROP, that demarcation line begins to take on a bit more body and becomes more like a ridge. And then in stage 3 ROP, that ridge begins to develop these finger-like projections that can extend up into the vitreous of the eye, which is just like the inside area there. And the problem there is that these blood vessels, when they bud up against the, that junction there, they'll begin to leak and bleed into the vitreous. And then eventually what will happen is they'll scar over, but when they scar, that scar anchors the retina to the back of the eye. And as the eye continues to grow, that's going to pull on and tear the retina and cause stage 4 ROP, which is here, which is a partial retinal detachment. And if that's allowed to continue for too long, then we get stage 5 ROP, which is a complete retinal detachment. And the retinal detachment essentially means that the retina is no longer on the back of the eye, and any light that's being focused by the cornea and lens is no longer properly being done, and you lose all visual acuity there in that region. But you can also imagine that having the disease, a retinal detachment, further out in your eye in the peripheral area is going to be less cause for concern than if you were to have it closer to the macula and the optic disc, which is why physicians also use this concept of zone to essentially determine where the disease is occurring. You might actually be more concerned about mild disease occurring here in zone 1 than having a little bit more severe disease occurring out in zone 3. And then finally, we have this idea of plus disease, which really gets at the way that the retinal vasculature looks. If the vessels are extremely dilated and tortuous, then we call that plus disease. We also have normal-looking retinal vasculature, and anything in between we call pre-plus disease. And it's combinations of these 3 clinical findings that actually alert us as to whether or not we need to treat a child, monitor them more closely, or if we can essentially watch them, bring them in for checkups every week or 2, or essentially discharge them. So in our moderate ROP group, or in the moderate ROP group here, these are children that need to be examined fairly frequently because they're at risk of progressing to our treatment-requiring phase of ROP. And these are children who have normal or pre-plus vasculature, but they have stage 1 or 2 ROP in zone 1 or stage 3 ROP in zone 2. Then in the treatment-requiring ROP group, these are children who also either have normal or pre-plus vasculature with stage 3 ROP in zone 1, or they have stage 1, 2, or 3 ROP in zone 1, or stage 2 or 3 ROP in zone 2, so long as plus disease is present there. So what you can hopefully kind of pick up here is that the presence of plus disease is actually a significant indicator of whether or not a child needs to be treated for ROP, and there's actually a pretty tight correlation between pre-plus disease and moderate ROP. So what we can do is actually kind of use these as the presence of plus disease and the severity of plus disease as kind of a latent variable for whether or not a child needs to be treated or monitored more closely. So our group actually developed this system that we call Deep ROP here, where essentially you can upload a child's retinal fundus image into a deep learning system here, and it will evaluate the retinal vasculature and produce a probability for that retinal vasculature either appearing as normal, pre-plus, or plus. But an issue with this is that this only works at the exact time of examination. So once you take that image of that child, you're uploading that image, and you can say, oh, this kid has plus disease, we need to treat them now, or they have pre-plus disease, we need to monitor them a little bit more closely. But it doesn't actually give us an idea as to who's on what trajectory. It would be nice to know if, for example, if you had a child who was on, kind of appeared in this area here of disease severity, whether or not they were likely to progress to plus disease or go to pre-plus, which would eventually goes away on its own, or if they were just going to remain normal over time. So that's kind of what the goal of this project to do, is to actually identify these children who are on these different disease trajectories. And we're going to do that, again, by evaluating the normal pre-plus and plus disease severities. And hopefully what we're really getting at here are the actual stages or the degrees of ROP here. And to do this, I'm actually hoping to kind of exploit this phenomenon that's present with ophthalmologists here, where if you were to ask them to, if you gave 10 ophthalmologists 10 images and you said, hey, provide me plus disease diagnoses for all these images, they would give you, it's almost guaranteed that they would all give you different answers for every single image. But if you ask them to rank the severity of plus disease here and put these images in order, they would all line them up pretty much the same way. So because that exists, that's what I'm hoping to exploit in my aims here. So aim 1A is to compute a nearest neighbors algorithm in a multidimensional space of subjects who are demographically and clinically similar to a given subject, and use the derived distance metrics as the weighted connections between nodes in a graph database. So the idea here is that we have this large data set from this imaging and informatics and retinopathy of prematurity multi-cohort study here. And this is from 9 study centers across North America. And all of the images in here, there's over 30,000 images that have all been graded by at least 3 trained graders. Some of the images have up to 9 different physicians evaluating these. And there are over 62 different clinical variables associated with all these patients here. So different things that set them apart from one another. Just some things to keep in mind here. This is a North American study, so it's pretty biased towards having a lot of Caucasian and white people. So while this will be a great pilot moving forward, if we want to implement this more globally, we'll have to keep some of this stuff in mind. But the idea essentially is that using all of these 62 clinical variables that are here, we want to plot all of our patients in this high dimensional space. And then once they're there, what we can do is actually perform a nearest neighbor search algorithm and compute the distance between every single patient or subject in our data set there. And then we can use a graph database then to essentially assemble all of that data, and the weighted connections between each of the nodes here will be the distance in that nearest neighbor search. And the reason we want to do that is because computing this nearest neighbor search every single time would be just unnecessary. We can just store those values once and be done with it. And querying this graph database is fairly quick and easy. And then once we do that, once we have a subject, what we can do is query the database, and let's say you had a subject who you wanted clinically was 750 grams at birth and a certain age. You could find subjects that were most similar to them, but who are on these different disease trajectories here, who are on a normal pre-plus or plus disease trajectory. And you could compare your patient's retinal fundus photo to these 2 other patients who are very similar to you but on the different trajectories. Now, to facilitate this, this is kind of where my aim 1B comes in, which is to develop an application that extracts subjects from this database who are most similar to the given patients and who are on normal pre-plus and plus disease trajectories, but then display their images to these physicians so they can more easily assess on which trajectory they fall. And to do that, I've kind of talked with some people, and it seems like the best idea here is to create an app for this, and I'd like to use what's called an Electron app. And this is basically many different things, applications that I'm sure many of us are used to using are like Slack, Atom, Skype, GitHub Desktop. They've all been built using this. And the beauty is that it's all just JavaScript and HTML and CSS, but then you can deploy it so it's on a desktop. It's pretty lightweight, it's cross-platform, and it has a completely open license. So we could actually get this to developing regions pretty easily. And the idea is that this would allow physicians to tune their patients' demographic and clinical features so that they could identify other patients who were similar. And once they did that, you could basically compare what your patient's image looked like right here to similar patients who are on these different disease trajectories, and you might be able to evaluate better where your patient was heading. But another interesting another thing here, though, is that although we're comparing patients who are very similar to one another, there's still going to be differences between them. So the real idea that we want to get at is, can we predict what your patient would look like if they were moving to plus disease, or moving only to pre-plus disease, or going to maybe regress back to normal? So aim 2 is to develop and train a deep convolutional neural network to produce images of subjects' retinas as appearing normal and having pre-plus and plus disease. Then we want to perturb and interpolate the latent space of that model to produce retinal fundus images displaying normal pre-plus and plus disease trajectories. So basically the idea is that if your patient's retinal fundus photo looks like this, doesn't show up super well on that screen, but looks like this, we want to predict what it would look like if it were normal, also if it had plus disease. And there's been some work around using deep convolutional generative adversarial networks to already create images, just making images from nothing, essentially. And the idea is that you feed in a noise vector into this convolutional neural network, and it compresses it into this latent space. That latent space is then upsampled to create an image, and that image, regardless of what it looks like, is then fed into this adversarial discriminator, which is essentially doing almost the exact same thing as this one, except once the image is compressed into the latent space, it's just evaluating whether or not that image actually looks like a retinal fundus photo here. But our real goal is not just to create the retinal fundus photos. We actually want to condition this, and not just the retinal fundus photos, but actually create those with a certain disease characteristic to them. We want them to appear, again, as normal, pre-plus, or plus. So to do that, we'll actually condition the latent space as well as the fully connected network in the adversarial discriminator. And to force this network to actually modify our images, what we'll do, instead of this noise vector, we can actually feed in our image here. And then on top of that, to force it to, or not even force, but I guess kind of promote it, to look more at the retinal vasculature, we're going to feed in these retinal vessel maps on top. And they don't show up well on that screen, but just believe that they're there. So basically, by adding this fourth channel, so most images are kind of a red, green, blue. They have red, green, and blue color channels. We can actually add this fourth channel as this retinal vessel map, and that'll help promote the algorithm to focus on modifying the retinal vasculature to look normal, pre-plus, or plus. Now, I don't want to spend the next year manually segmenting these retinal vessel maps by hand, so my plan here is to instead train an encoder-decoder network here to perform these segmentations for me. And the idea, basically, is that you feed in a couple hand or manually segmented vessel maps there, along with the original image up here into this network. And the idea is that it compresses it down into this latent space, and then it tries to regenerate that image. But when you're doing it, it's actually performing pixel-level labeling. So instead of just trying to recreate this original image over here, all it's doing is saying, for every pixel in this image, what's the probability that this pixel belongs to a retinal blood vessel? And if it does, then it'll give that probability 1. If not, it'll be 0. And in these images, a 1 appears as white, and a black appears, or a 0 appears as black. So once we have these images formed, the idea then is, so we'll be evaluating these images and looking across these, this GAN architecture, and we'll be using generative adversarial loss to evaluate how well the model thinks it's doing. But eventually we need to actually have people, like humans, look at these images and say, do these even look like retinas? And if so, are we getting the disease characteristics that we were expecting? Does a normal retina look normal? Does one with pre-plus look like it has pre-plus, et cetera? Or is the image just completely undiagnosable? Now, assuming that this all works well, the idea would then be to kind of interpolate across the latent space of these. So I keep talking about the latent space there. And so that's just basically this compressed representation of our input image, meaning that all of the important features that made up that image have been reduced into this smaller set, and then all of the extra information is just that. It's just extra, and it's not actually necessary for reconstruction there. Now, in the original Deep ROP paper that was released, Dr. Brown actually performed a t-SNE, used t-SNE to evaluate the latent space of the model here. And basically what this is showing is that this is basically the high dimensional space of the CNN, but it's just down into two vectors here. And what you can really see is that there's a clustering of normal eyes, of pre-plus disease eyes, and plus disease eyes. And you can actually see that there's kind of this natural continuum here of disease severity. So the idea behind our model is that if we're generating images, if we can generate an image of your eye that looks like it's normal and an eye that looks like it's pre-plus, then we can actually generate the images in between here and theoretically show you what a natural progression from normal to pre-plus would look like. Or we could actually show you what normal to plus disease would look like across this continuum here. And if that were to work, then we could actually integrate this into AIM-1. So instead of just having your similar subjects down here, you could click on another button that would predict the trajectories here and instead show you for your patient what a progression to normal, pre-plus, or plus would look like, and maybe what you could expect your child to look like at a next follow-up visit there. So that said, there are some limitations to this. So the first is going to be the data set size and the demographics that I kind of mentioned earlier. So we can perform some up and down sampling on the underrepresented and overrepresented groups, respectively. We're also in the process of acquiring data sets from other regions. So we do have our hands on a data set from the Aravind Eye hospitals in India. But also with these deep learning models, you can perform these random flips and rotations and zooms on the images to sort of synthetically augment your data set. And that's because these models are typically, these deep convolutional models are invariant to these sorts of changes in an image. So you and I might know that an image is just an image rotated 5 degrees, but a CNN says, oh, that's a completely different image. So we can synthetically modify our data set to go from maybe 30,000 images up to 300,000 if we wanted to. The second thing is that we're going to use this model, we're going to use some pre-trained networks, or pre-train our network at least, on the ImageNet database, which is a database of over 14 million images there. And that the idea there is that it'll at least learn some basic shapes and of everyday objects prior to learning on our data set, so it doesn't overfit. We'll also be implementing dropout and max pooling within our model here. And then we'll be performing some dimensionality reduction on our images. And also we're constraining that feature space by forcing our model again to kind of look at the retinal vasculature. Another problem here is the inter-rater disagreement. So as I mentioned earlier, if you give 10 physicians 10 different ROP images and you said, hey, which of these have plus, pre-plus, or normal vasculature, they would likely grade those differently. Our attempt to address this is we have at least three ROP experts grading each of these images. Some of the images have up to 9 people grading them. But instead of just taking a majority vote, we're actually using a consensus diagnosis here. So anytime there's a disagreement about what a diagnosis is, there's a conference that occurs, and people are essentially trying to talk each other into why they're right about the diagnosis there. So at least you can get everyone to kind of agree there. And then finally, there's this issue of model interpretability with deep learning models. And that's kind of why what I'm hoping to do is this whole image generation and latent space interpolation here. The idea is that, if I could easily, not easily, but I could make a model that just produced gave you a probability of a child developing plus disease at some point in the future, or developing pre-plus disease. But there's a lot of pushback from physicians about these models because they're kind of deemed black boxes, and you're not exactly sure why that's occurring. So in this sense, we're going to be using a black box to generate the images and at least lend to some interpretability, show physicians the images that we expect, that we might expect a child to progress to or regress to, and hopefully that empowers them a little bit more and they're still able to make an accurate diagnosis. So the implications for this essentially are that access to ROP resources it's just going to be a problem for the foreseeable future here. So if we can just streamline the ROP examination process and route the currently available resources that we do have to those 15% of children who need examinations more frequently, who need to be treated, then we can essentially triage them better. The low-risk individuals could be examined less frequently, and hopefully that this would just decrease the incidence of ROP-related visual loss worldwide. But outside of ROP diagnosis, this has many other possibilities here. So we could apply this to other ophthalmic diseases. We could also apply it to other imaging modalities within ophthalmology, but it could be interesting to apply these to completely new fields with completely different imaging modalities altogether. So with that, I'm going to conclude, and I will happily take any questions. Yes.
Mm-hmm. Question about the decision-making piece of this. So, oh gosh, this goes back 15, 20 years ago. I did a paper where we were looking at diagnostic accuracy. Mm-hmm. And one of the things that we learned was, in talking with experts, that anybody that had a, quote, suspicious biopsy went on to surgery. So what currently happens to these images that they don't really know about? Do they go on for treatment, and is there a downside to overtreating?
Can you, sorry, when you say images that are suspicious?
These were biopsies. They weren't images.
Oh, okay.
So, but there was disagreement among the pathologists about whether it was cancerous or not.
Okay.
So those people often went on for surgery.
I see.
So in your situation where the images, there's images that may be unclear or suspicious, what happens in practice to those?
I'm not completely sure what happens. Pete might be able to answer a little bit more. But my thought is that you would either wait a little bit longer until the disease either progressed to, so you could make a very clear diagnosis off of it. I know that there are some people are looking into even like other early treatment. There's already been one early treatment study that's gone on, and it's actually been pretty successful. But even further, the idea would be if you can identify children who are going to eventually progress to plus disease, if you could possibly treat them even earlier.
So is there a risk to treating someone that maybe didn't need to be treated?
I would assume that there definitely is, because the treatments are essentially cryotherapy or laser ablation. There's also an anti-VEGF injection, but you're scarring the retina.
Okay,
so if you're unnecessarily scarring it, yes,
okay,
there could be some problems.
That's what I wanted to know. Thank you.
Yeah.
So I'm just curious what demographic or variables from the demographic variables from the EHR are you including in your graph database, and will you observe what variables have the greatest influence?
Yeah, I'm thinking, so for that database, there's all sorts of patient information. We have race, ethnicity, gender, how old you were when you were born, how much you weighed when you were born, what your current post-menstrual age is, all sorts of different things. That is something that I'm actually, so I was kind of thinking about building a model that just includes a lot of that stuff, but then I'd also like to go back and kind of trim and find out what's actually necessary within there. because there's a lot of stuff that could sway. You know, as I mentioned, like this is a very North American database here, so we don't want to just be saying, oh, everyone's the same because of one factor or another there. Yeah.
Hi. I have a related question about that first aim. And it's kind of a two-part question. So the first is there literature that suggests that ROP does present differently or appear differently in people from different demographic or clinical backgrounds?
I don't know exactly. So I think the idea would be, what we're really trying to do is capture patients who are at that same clinical time point. So it's not necessarily that if we have someone who is Asian versus someone who is white that we're trying to, you know, they're completely different. But it's more about, you know, if you were born at 36 weeks and someone else is born at, or not 36, if you were born at 31 weeks and someone was born at 26 weeks, you know, even though you might currently have the same age after birth, like you're not actually the same, right?
Right.
So we're trying to line up a little bit better there. It might be that we don't need to include as much of that demographic information.
I wasn't suggesting
Yeah,
that.
no, yeah.
But, so it sounds like it's largely about the developmental staging and developmental age of the child that you're trying to capture with this.
Yes.
That's great. And I was also wondering, related to that, do you have a plan for how you're going to evaluate the success of that first aim? Like, how do you determine if the graphing approach and the network approach is telling you something meaningful?
Yeah, so that's something that we're actually kind of discussing, you know, because, yeah, just because we present those images doesn't mean that anything is going to be lining up the same. And it's very possible that, you know, your children who look like pre-plus are going to be all over the place, even if they're at that same developmental age. So that will be something we're going to have physicians kind of look through, and we're going to actually assess those, you know, assess each of those and see like what the accuracy is of using that model to predict that. And that's kind of where that second aim comes in. Also, like if that doesn't work, then maybe predicting what you would look like at a certain point will be a little more informative. Yeah.
I have a question about the trajectories. If you start to have clinicians who want to put multiple inputs into that, meaning like they didn't know about your resource until they'd actually taken, you know, a series of fundus photographs over time, or they have a couple of different ones that have maybe different quality parameters, do you have a plan for what happens when people try to input a series, whether it's a chronological series or multiple images of the same thing, in terms of how you would deal with, you know, kind of showing a progression if people are submitting things that are already a progression?
Yeah, I have dreams for that. Let me find that. I proposed this to something like this to my DAC. So the idea with this is kind of this predictive conditional GAN here, where you could actually upload a series of images, and then it would produce what the next image in the sequence would look like, theoretically. So that way, if you had just like that, you didn't have all this clinical data, but you had a bunch of images, you were curious where your patient's likely heading, you could actually predict that next fundus photo. That would take a lot of data, and I've been told that my aspirations are very high, so yeah. Yeah, that next one. Yeah.
So I had a similar question about kind of progression. I was wondering if the database you have access to has longitudinal data.
Yes, it definitely does.
Okay.
Yeah.
And then one question, I'm sorry if I missed this and you talked about it, but when you talk about generating images to show a progression over time, are you able then, or could you leverage that database to take the first one and then try and progress it over time and see if it matches up with what the actual progression
Yeah, so
was?
that would be the idea, actually,
Okay.
right? So you would take that progression and you could say, oh, well, if my child's on a progression to plus disease or pre-plus disease, then this is what they should look like by the next imaging session or the one after that, right?
Yeah, I guess my question was, in your generation of images, are you planning to use actual progressions as like a gold standard to measure against and say, oh, our image generation is actually doing a pretty decent job?
That's
or
interesting, yeah.
for training purposes?
I hadn't necessarily thought of that. So one of the issues with this, so like this idea would be taking your images that you currently have and generating what your image would look like. The other idea that I'm kind of proposing here is that it would take your retinal fundus and modify it to look like it had plus disease, but it wouldn't necessarily be exactly what plus disease is going to look like in your eye. Does that, the retinal vasculature will be just as severe, but it might not be exactly the same.
Mm-hmm.
So matching up those images one-to-one might not be the best method there.
Okay.
But if you could look at severity between the two and say, oh, this one's just as severe, then yeah.
Actually, I think this is somewhat along the same lines. I mean, first of all, I think the AIMS 1A and B are fantastic. I think that a lot of the machine learning imaging studies now are, basically based on single point in time, and it sounds like you're adding a temporal component, which sounds wonderful. I guess I'm trying to think of the clinical value of the simulated images, given already the lack of complete trust in black boxes that physicians have, and wonder whether it might be more valuable to those physicians to actually just see what those other patients that are matched through the graph database and the temporal and then the disease trajectory, saying, here's like, I could imagine you showing on a screen, 9 different images of real patients, and you might see the spectrum as opposed to sort of trying to simulate something because physicians, I think, may be a little skeptical of that.
Yeah.
So I wonder if a second AIM, might be more focused on, instead of telling the physician this is some simulation of what your patient might look like, instead, here's a representative sample.
Of what those.
Yeah, I guess it comes down to what the physician is going to do with the images that they see.
Yeah, yeah. I mean, that's not a bad idea either. I guess the idea with generating the images is that you could actually show your patient a representative sample of what their patient was going to look like. You know what I mean? Like at that certain time point. So it might just be, I don't know. It's interesting. Yeah.
But I think what you're suggesting is already in your AIM 1, right?
Correct. I mean, to a degree, right?
It's
We're going to show-
AIM 1 is sort of picking from a database, right? Picking from thing, and then AIM 2 is actually sort of unraveling the actual underlying latent structure so that you can do this style versus content separation types of things that GANs are so good at, essentially. I mean, I agree, sort of like maybe physicians will never accept machine learning, but it's our job to keep trying.
No.
Until you guys give up.
I agree. I just think that, I see it sort of in AIM 1B. I'm just thinking as the next step, whether sort of saying, here's a spectrum of real-world cases. But I don't know. That could be the K Award after he gets his PhD.
How many images do you eventually expect to get? Because that could completely make or break this deep learning approach.
What do you mean? Eventually expect to
Your
get?
data size.
I think we're looking, so I know this one has at least 30,000. There's up to, I think, 36 now. It's continually growing. We also have the data set from the Air-Vent eye hospitals, and then we're also getting another data set in here. Pete, do you have an idea of the total size here?
We're starting a prospective study in India which will enroll 3,000 babies per year. Each of those babies will have a number of images at a time.
Yeah.
We have access to retrospective images from that data set, which have something like 50,000 right now.
Yeah.
That's an enviable size for most medical imaging related questions.
And we have,
But on another note, since what you really care about in terms of prediction or trajectory is the severity,
correct.
That's one variable. Continuous or binary, doesn't matter. why don't you just aim for that instead of having to project a new image, which is in a much higher dimension?
Because it's more fun? I know.
Yes,
So, I mean, that is definitely something that we could do. And like, that's kind of like a backup plan, right? That you could, instead of just going through all of this and trying to predict the image, you could actually just say, hey, this is what's going to happen eventually. But I think this just lends a little bit more to being able to, you know, kind of actually look at the natural progression of your child. And it, in a way, makes it so that physicians everywhere can just kind of actually understand what's going on and see where their child or where their patient's heading. So it just lends to the interpretability a little bit more.
Sure. I have a question and maybe a follow-up to what Bill is saying. So when you're planning on projecting these images, what time scale are you looking at?
Yeah, so that's going to be another interesting thing that we have to kind of figure out.
So I would, so getting back to what Bill is saying, I would really think about, you know, the clinical decision points and what time scale is actually useful.
Right.
And if the decision, I mean, what I got from your earlier talk was that, you know, the decision about how closely do we have to follow this person, do we need to see them next week, do we see them next month?
Right.
You know, I think it'd probably be more clinically useful if you projected these images along these different trajectories, but just in that next decision time period. Mm-hmm. So if they went in the pre-plus direction, what would they look like in a week? If they went in the plus direction, what would they look like in a week, et cetera, et cetera.
Right.
Now, and maybe all those images look exactly alike because they're not going to progress very much, or
right.
maybe the images are going to look very, very different, and then you have something that you've given the clinicians to say, well, you know, if they go in the plus direction, it's going to be a lot in a week or in 2 weeks, and you know, give them, I mean, you are trying to predict the future, but I think that's the clinical problem, right? When do they have to take action, and when can they take action early enough? So thinking about, you know, not just, you know, what would the images look like in the long run, which I don't think is actually all that clinically useful, but what is the increment, what do the images look like in a time increment with which I need to make a decision.
Right, and that
might be actually an easier problem depending on if you have the data, because it's a smaller extrapolation.
Right,
and also might be more useful.
Well, and that's why the idea is there. Once you can actually generate those images, then to interpolate across that latent space there. So then you can get those images that are in between at those different weeks. You know, so you're not just we're producing the end goal, but what does it look like as it's going across there? And we would have to figure out, you know, what are the different time increments across that too.
Any more questions?
Actually, let me just add one comment, and admittedly slightly toot my horn. There's actually a really nice paper in this week's issue of New England Journal of Medicine about machine learning in medicine. One of the co-authors is Zach Kahani, who's the head of informatics at Harvard. The little toot is that one of the papers cited in that paper is a paper that I did with some colleagues. But despite that, it's actually a really well-written and readable article about machine learning in medicine. So people might want to take a look if you want a kind of big picture about machine learning in medicine. Okay, thank you. We'll look forward to your defense.